Okay, so this is the keynote lecture. So I try to give you an overview. It's not on the last result of my lab, which are always, I hope, exciting. So it's a bit more global. I changed a bit the title to put parasite because I will speak a bit more about parasites than bacteria and fungi. So flies live on rotting fruits. This is an ephemeral ecosystem, so they have a short lifespan because they need to adapt to the short life of their ecosystem. And I should say that bacteria and yeast are part of a source food for them. So they live in a very microbe-rich environment. Flies and Drosophila fight many pathogens. It's a bit like us. You see they belong to the same classes: virus, bacteria, fungi, and parasite. And they have developed a kind of complex way to deal with microbes. So usually we define host defense mechanisms at broad in 3 dimensions: immunity, which are involved to eliminate the pathogens, and I will come through all those mechanisms briefly. Disease tolerance, that was discussed by Miguel, are mechanisms that do not target the pathogen but allow to resist infections, so stress response, wound healing, metabolic adaptation, detoxification. And then I will mention sometime behavioral immunity, which are mechanisms that allow, for instance, insects but also humans to avoid to be infected or to clean, like to remove the spore or to stop to eat when you get infections. So what I was planning to go there is to go through the Drosophila immune module, so it's a bit of a generic talk, try to give you a few insights about what we have learned in the last years and sometimes difference with vertebrates. So what are the mechanisms that use Drosophila to fight against virus? So the main mechanism that you find also in plant and in nematode is RNAi. And in this case, this is both an innate and adaptive immune mechanism because you have dicers that recognize double-strand RNA from virus and will generate some very small siRNA that will target the virus. So this is extremely specific. The importance of RNA is shown because many viruses that infect insects have inhibitor of RNA. Finally, work done by Carla Salé and others have shown that you have long-term antiviral response. That means when you get infected by a virus, this lasts for a long time. The second mechanism which is used to fight against virus is a bit more insect-specific and surprising in some way. I need to tell you what are endosymbiotic bacteria. So many insects are infected by vertically transmitted bacteria that go from mother to offspring. Their names are Wolbachia, Spiroplasma, Hamiltonella, and probably the most successful intracellular bacteria is Wolbachia, because these bacteria can infect 40 to 60% of all insect species, which means more than 1 billion of hosts. Wolbachia is inside, it is quite globally silenced. And work done by Luis Teixeira, when he was postdoc in Cambridge, he realized, after many studies, that flies that contain Wolbachia are more resistant to viral infection. And the level of resistance is quite similar to the level of resistance that you find with RNAi. So here you see an example of a tripartite interaction between a bacteria, a symbiont, that protect the host, an insect, against a virus. And you understand that if you treat flies by antibiotics, you make them susceptible to viral infections. And so you understand also that by acquiring one bacteria from the external, you can gain a very strong resistance against virus. The mechanism is not very clear. I will speculate that this has to do with lipid competition with lipid droplet, and other aspects that are important for virus. Now, there are other antiviral defense in Drosophila, but they are not virus, they are virus-specific. There is no evidence of type 1 interferon response, a kind of generic antiviral response in Drosophila. But there are in the genome many loci which are involved, which are refractory loci that block virus infection. And here maybe because flies has a short span life, because it has a very broad polymorphism, can easily pick up maybe mutation that confer resistance to virus. Now I move to a second aspect of immunity, the gut, or epithelial immunity, which is very popular those days. And of course, this is the main route of infection, like in us. So what do we know about the gut? First, what is difficult for microbes with insects is that you find chitin everywhere. So it's very difficult to enter. Even in the gut, you find a chitinous matrix called the peritrophic matrix that prevents bacteria to touch the gut cells. The consequence of that is that you will not find in insects invasive bacteria such as Shigella, Salmonella, or Listeria, because the bacteria cannot interact with the epithelial cells. The mechanism of pathogenesis is bacteria that produce toxin that can cross the peritrophic matrix and will damage the whole cells. And here you can see that there is a trade-off between permeability of these barriers that allows the digestive process and cross-linking of these barriers that allow to prevent the action of performing toxin. And we have been able to show that. Acidity, digestive enzyme, peristaltism are believed to be important to fight microbes. One of the first immune reactions that you find in the gut are production of reactive oxygen species by two enzymes, diox and NOx. Interestingly, one of those enzymes has been shown to be activated by uracil that will bind to a G-couple receptor, activate a pathway that releases calcium, and activate this enzyme that produces reactive oxygen species. Finally, I just want to mention that the fly has also a gut microbiota. It is a bit more inconstant because there is a constant transfer of the microbe from the outside from the inside, and it has been shown that this microbiota affects the host-pathogen interaction because it stimulates basal immunity. So one mechanism that I want to speak a bit more is the antibacterial response that takes place in the gut. So this is shown here. This is mediated by a pathway which is called the IMD pathway. So this is very simple. Bacteria release peptidoglycan. This is a molecule which is recognized by a receptor called PGRPLC, peptidoglycan recognition protein LC. This activates a pathway which I will not describe, and this leads to the production of antimicrobial peptide. Interestingly, the lab of Julien Royer and our lab, we could show that in the middle part of the gut, where digestion takes place, where you find this peritrophic matrix which is very much permeable, the fly use an intracellular pattern recognition receptor called PGRPLE, and this sends monomeric form of peptidoglycan. We believe that this mechanism is important because it prevents a kind of presentation of the pattern recognition receptor in an environment where you have too much microbes. So you see here one feature about the gut, that this immune response is very much compartmentalized. And we should not call the gut immune response, but each domain of the gut has its own immune response. One aspect which is important with the gut is that immunity is just one part of the story. And we could show that here you see a gut of a fly, non-infected, but you see that 4 hours after an infection with a bacteria, the size of the gut is half of the size and very thick. Why? Because half of the cells, enterocytes of the gut, will die and delaminate. But interestingly, 2 days after, the gut is completely renewed, exactly identical. How this is possible? This is possible because you activate the stem cell renewal program. So globally, what we could show is that bacteria enter through the gut. They are recognized. They trigger the production of antimicrobial peptide that kills the bacteria. They trigger also reactive oxygen species that kills the bacteria. But the side effect of that is to damage the epithelial cells that start to delaminate. And we are able to identify some factors which are released by the cells that delaminate to inform stem cells to divide and to repair the gut. So you maintain barrier integrity. If you will guess what is the most important, is it immunity or epithelial renewal? In fact, in the gut, epithelial renewal, which refers to, in fact, disease tolerance, if I come back to the previous talk, is more important to survive infection. Now I will move to what is called systemic immunity. So systemic immunity refers to immunity in the body cavity of the flies. So what's happened when you infect flies in the body cavity? So one of the most popular and characterized immune response is the production of antimicrobial peptide. So when you inject bacteria in the body cavity, you activate a number of pathways that lead the fat body, equivalent of the mammalian liver, to produce a battery of antimicrobial peptide. The first feature that you observe is that those antimicrobial peptides have distinct activity against either fungi, gram-positive bacteria, and gram-negative bacteria. Antimicrobial peptides are found in all animals and also plants. They are universal mechanisms of defense. But in humans, for example, they are produced in mucosa because you need to reach a high concentration. But because Drosophila have a small volume, you can produce those antimicrobial peptides to a high titers. So this is something specific of small insects. In big insects, you will not see systemic production of antimicrobial peptides. So work have shown that this response is regulated by two pathways, the TOL and IMD pathway, which are activated by different classes of microbes. So you see gram-negative bacteria are sensed by peptidoglycan that bind to a transmembrane receptor, and this leads to the activation of the pathway. In contrast, gram-positive bacteria, yeast, are activated by secreted pattern recognition receptors that bind to the microbes, activate a cascade of serine protease, cleavage of Spätzle, activation of the toll. All the intracellular part is conserved with mammals, they are a kind of NF-kappa-B-like pathway. So you would say, why do you have this architecture of those two pathways? Because Drosophila do not sense lipopolysaccharide. It has not the equivalent of toll-like receptor, which is maybe clever. It can sense only peptidoglycan, and peptidoglycan is released when bacteria divide. So what the immune system sees is bacteria that divide, which is a signature of infection. And so here you have an indirect mode of recognition because you recognize the bacteria by the release of the elicitor. In contrast, gram-positive bacteria and yeast harbor their peptidoglycan, their pumps, at the external layer and can be sensed by secreted pattern recognition receptor that can bind to their surface. So you get a different type of architecture. Now I will speak a bit more about, because we speak about evolution, shaping, about peptidoglycan recognition protein. So what are PGRP? Those are proteins which are involved in the sensing of peptidoglycan, and they function as pattern recognition receptor in insect. So here you see the structure of a PGRP. It is similar to T7 lysozyme, which are enzymes that cleave peptidoglycan. And in fact, you find two types of PGRP. PGRP, which are enzymes, they can cleave peptidoglycan. But you find another class of PGRP which can bind to peptidoglycan but are mutated in their catalytic site, and they are used as pattern recognition receptor. So first intriguing observation of Drosophila, which to my knowledge is not observed in mammal, maybe perhaps CGAS, but that these fly use molecules that were used as enzyme as pattern recognition receptor, okay? And they evolve later as PGRP. Here I show you a beautiful picture of pattern recognition receptor in Drosophila. This is peptidoglycan that you can see, which here it's a polymeric. It can cluster, bind to different PGRP domain, and this bring together the cytoplasmic of PGRP-LC and activate the IMD signaling pathway. If you activate a immune response, you don't want to activate a too strong immune response. And we have shown that negative regulation is very important. And one interesting negative regulator of the immune response is called PGRPLB. So you see the gene PGRPLB is activated by the IMD pathway, but PGRPLB is an enzyme that can cleave peptidoglycan in some fragment that cannot anymore activate the immune response. So it can shut off the immune response. So you see here, bacteria divide, release peptidoglycan bind to the IMD PGRPLC, this activates the IMD pathway, production of antimicrobial peptide, but at the same time, production of PGRPLB that can scavenge peptidoglycan and shut off the immune response. More recently, we have went a bit further. We have identified another PGRP called PGRP-SD, which is also regulated by the IMD pathway, but this is not a catalytic PGRP. And this PGRP can bind to peptidoglycan and will bring peptidoglycan in close proximity to PGRP-LC. And what you see is that the IMD pathway is regulated by positive and negative regulator. What was a bit surprising is to observe that double mutants that lack PGRP-SD and PGRP-LB behave like the wild type. And you could wonder why to develop those negative layers. If you remove them, you get. But what is interesting is that if you, by modulating those genes, you can modulate the immune reactivity of the flies. And this gives lots of flexibility to, for example, make a tissues of an organ much more sensitive. You don't touch to the core of the pathway, but for instance, by increasing PGRP-SD, you will make the flies much more reactive in terms of immune response. So all that shows that the IMD pathway and immune pathway can be adjusted to the severity of infections. So here I give you a little speculation about the evolution of PGRP. So bacteria, peptidoglycan, is a source of food for Drosophila. And Drosophila is equipped with a large repertoire of peptidoglycan interacting protein. 30 genes encoding PGRP, but also 13 lysozymes. I just want to highlight that bees or silkworms that live on very clean food have only 1 or 2 lysozymes. Among the two, it seems that PGRP have acquired multiple functions. And we could speculate an evolutionary scenario for them. They were first digestive enzyme. Maybe later they were involved as antibacterial factors. Later on, they were used as pattern recognition receptors that can be used as pathway. And later on, some of them were used as immune regulators, such as PGRPLB in PGRP-SD. Now I want to come back a bit more to an interesting observation that was done in the lab of Julien Royer in Marseille, which referred to behavioral immunity. So they observed that when flies are infected, they stop to lay eggs. So they keep their eggs, they have a kind of stopping oogenesis, and we could speculate that this is a mechanism used to fight or to better against the infection and stop because of a trade-off oogenesis. And interestingly, it could show that this response is also mediated by the IMD pathway, but in this case, in neurons. So when you get an infection, peptidoglycan is released, it goes to the fat body to activate the antimicrobial response. But peptidoglycan is also sensed, in this case, by the intracellular PGRPLE that lead to the activation of octopaminergic neuron, and this leads to retention. So we have here an example of an immune pathway repurposed for a behavioral response. And we can speculate that many pathways which are used actually to sense microbes are also used to sense microbes in terms of the neuron system. So here I speak a lot about Drosophila, but I just want to show you that insects are very different. And here you see an insect that you don't like so much. It's a plant pest. Huh? Those are aphids. And people have worked on and so aphids feed on sap. Sap is just sugar. It's a very sterile food. And people have looked to their immune system, and you see no IMD pathway, no PGRP, no antimicrobial peptide, no lysozyme. You find some hemocyte, some melanization, and interestingly, you find endosymbiont-mediated protection. I just go briefly on that. Here you have an aphid infested by a parasited wasp. I will come back to that. But this aphid is contained not by Wolbachia, but another bacteria called Amyltonella that produce a toxin that kills the wasps. So they can use endosymbiont to resist infection. But you can see here that relaxed selection for the maintenance of costly immune function, these aphids have lost a large part of the repertoire of the human genes. Now I will move to cellular immunity. Before to move to cellular, I'd like to remind you that one of the protagonists of cellular immunity did this work on the starfish, and he was not the first to analyze phagocytosis, but he was one of the first to realize the importance of it, and this also highlights the importance of invertebrate model. So what do we know about cellular immunity in Drosophila? So there are, interestingly, only 3 cell types in Drosophila, which are involved, each of them, in different immune function. You find plasmatocytes, they are involved in phagocytosis. Crystal cells are involved in melanization. I will come back to that later. Lamellocytes are large flat cells which are involved in encapsulation of parasites that cannot be phagocytosis because they are too big. The classical view of hematopoiesis was a kind of binary switch between a number of transcription factors. But recently, a number of people, and I don't have time to list them, have shown that you have many evidence for transdifferentiation. That means the hemocyte or cellular lineage is much more flexible than what we expected. Plasmatocyte can change in crystal cells, and plasmatocyte can change to plasmatocyte. So what do we know about hematopoiesis in Drosophila? And here, this is something that I want to highlight, is that also there is strong trade-off about the production of hemocyte, and hematopoiesis is something which is very costly for the host. So in Drosophila, hemocytes are made at the embryonic stage. They colonize the larva, and you find them either in circulation or in periphery. So in Drosophila, we use the term sessile hemocyte to mention hemocytes which are attached to the cuticles. Okay? So they are a bit like resident macrophages, if you want. Interestingly, there is an organ called the lymph gland that are used as the reservoir. So you have lots of hematopoiesis going on in the lymph gland, but the lymph gland will break only at the metamorphosis to release more hemocyte, or if there is an infection by a parasite. So you can see here the organization of the immune system in 3 compartments: a storage organ, sessile hemocyte, and circulating hemocyte. So recently in the lab, we have identified a mutation that lack all the sessile hemocyte. So this is a mutation in an EGF transmembrane receptor called ITER, and you can see here, those are flies that contain GFPs that are expressed in hemocyte. You can see that you don't see anymore hemocytes which are sessile attached to the cuticle like in the wild types. And so we are now trying to understand what are the role of sessility, why hemocytes need to attach to the fly. And I don't have time to go too much in detail, but it seems that this is a way to control hematopoiesis and also to stop hemocytes to divide. And here I want to highlight that there is a kind of trade-off. More you have hemocyte, more flies are resistant against parasite. But more you have hemocyte, more flies are susceptible when you put them on a poor diet. That means that the number of hemocyte blood cells need to be strongly constrained by the trade-off between pathogens and also survival on poor diet. So studies have shown that hemocyte has multiple roles. They are involved in tissue remodeling, production of basal membrane, phagocytosis of apoptotic cell, the systemic wound response, production of antimicrobial peptide, phagocytosis, storage. So they are very important to storage of a number of enzymes. They contribute to the clot that you can see here, and they are involved in sequestration of parasite. A new concept that has emerged in the last years is that hemocytes are local repairers. That means they are not just immune, but they go to tissue which has been wounded like that, and they can release a number of morphogens like DPP, hedgehog, that can participate to the repair of the gut. So as soon as you get a damage somewhere, hemocytes start to come and can contribute externally to a number of growth factors that will allow to regenerate. So now I have been to all the mechanisms, and I will focus a bit more time on encapsulation. So here, so I need to activate this little movie. Okay, so what do you see here? You see a parasitoid wasp. This is a large class of insects. Think there are more than 100,000 species of parasitoid wasp. And this parasitoid wasp is like a cuckoo. It is a bit lazy. It lays its eggs in the larva. You see, it has a kind of machine that allows to lay its eggs in another insect. And those, of course, you understand that this parasitoid is completely dependent from the host. And now what I want to discuss is what are the mechanisms used by the larva to fight against this parasitoid wasp. Here you have a fight, insect against insect. The first question that emerged is why larvae usually lost the combat against parasitoid wasp. It's because parasitoid wasps are specialized predators. All their life rely on the other, while the larvae are infected by many more pathogens and allocate only a part of its resource to fight against the parasitoid wasp. This is why parasitoid wasps are used in many places to fight against insects. You release the parasitoid wasp, and like that you can block an insect because you block the growth of this insect. So now, okay, so I try to, okay. So this wasp lays an egg in this insect. You see that this egg is inside. It will start to eat slowly the insect from the insect. Now you have two outcomes. The first outcome is that the insect, here it was a Lepidoptera, a butterfly, can encapsulate and kill the wasp, and the insect wins, the Lepidoptera. But in most of the cases, the wasp will win, and you will see a new wasp that can now infect another insect. So they are called parasitoid and not parasite because only one can win. You never see the two together. So parasitoid wasps are extremely potent. They do not inject only their eggs. They have also a gland, a venom gland, where they will inject many molecules that will suppress the immune response. So they inject many, many virulence factors that will block the capacity of the flies to encapsulate. So what are the mechanisms used by Drosophila to fight against those wasps? And here you have the problem of the parasites. That means the insect needs to recognize another insect. So what we observe is that as soon as the egg is injected, it is covered by hemocyte. Hemocyte attach, and in the base case, you have formation of those lamellocytes and a capsule which is melanized, and the flies can kill the wasp. We have not a clear idea of how the wasp is recognized, but the concept here is probably recognition by absence of self. The idea is that the parasitoid will be recognized by hemocytes because they are not protected by the basal membrane. That means all the tissues of the flies are covered by basal membrane, and hemocyte has learned when they touch the basal membrane to stop and react. But as soon as you put a tissue which is not covered by the basal membrane, they start to encapsulate. Okay? Finally, studies have shown that the protection correlates with hemocyte number. But I just show you that the number of hemocytes is also linked, as also a trade-off because hemocytes consume lots of energy. Another interesting mechanism used against parasitoid wasps that we have discovered is the case of spiroplasma. Alors again, here I speak about an endosymbiont. You see, it's a bacteria which is a bit strange, called Spiroplasma pulsoni, that can infect Drosophila and is vertically transmitted from mother to offspring. More recently, we and other labs have shown that the presence of Spiroplasma can block the success of the parasitoid wasp. two mechanisms have been proposed. We have proposed that this is competition for lipid. Spiroplasma will take the lipid that the wasp need. But another group, and I think I start to believe more in their study, have suggested that spiroplasma produce a toxin that will directly target the parasitoid wasp. Here again, by acquiring an external bacteria, the insect can fight better against the pathogens. Okay. Finally, flies have also developed many other mechanisms of behavioral immunity to fight against parasitoid wasps. And here it's a beautiful experiment where it is shown that flies that smell parasitoid wasps, so when you put parasitoid wasps, they can feel it and they start to lay their eggs in medium which are rich in ethanol. And why? Because wasps never go to medium which are rich in ethanol. So the idea, you see the title, fruit fly medicate offspring after seeing parasite. They can lay their eggs in a medium which is more adapted for their progeny because there is less chance that parasitoid wasps will come and infect their progeny. So we show you that behavioral immunity is extremely sophisticated. So now I will do a little summary of the data before to move to another point. Drosophila is one of the best characterized immune systems among metazoans with human, mouse, zebrafish. Everything is innate, so it depends what you call adaptative, but there is no kind of rearrangement of somatic gene like we see in mammals. But there is evidence of long-term or persistent response and also evidence of specificity. It is composed of different modules that can be deployed in a coordinated manner against distinct pathogens. I show you few of them, but each of them are activated again. The immune response is compartmentalized. You have local and systemic immune response, but also interaction between the two. I show you that in insects, symbionts contribute to our survival. I speak about Wolbachia against virus, Spiroplasma against parasitoid wasp, and I mention the role of gut bacteria that stimulate the basal immune system. I show you that hematopoiesis is more dynamic and flexible than we see and can really adapt a lot. And finally, host survival is not just a question of immunity. I show you the role of stem cell. Of course, the fly immune system is shaped by its ecology and the pathogen found in the environment. And this is the case with the aphid that have lost a large part of the immune system. So now I want to tell you in the last 5 minutes what we are doing in the lab, like that a bit more. So what has limited our understanding of Drosophila immunity? We have focused on the same question. There is a kind of emphasis on what is conserved, which I find is a mistake. Most immunity genes belong to large gene family, like more than 20 antimicrobial peptides, 12 PGRPs. Many immunity genes have a subtle phenotype. And this is a problem for the single gene approach. Many of the large-scale approach, such as in vivo RNAi, have led to many problematic studies. I won't go to detail, but they have been a bit disappointing. So the main problem was the difficulty to generate mutants. And now we can make mutants very easily. So I have the chance to have a collaborator from Japan that sent me many, many mutants. And I think that with about 200 mutants, I will have what I need to understand the fly immune system from a genetic point of view. So what we are doing in the lab is the following. We now target gene family using mutants, but we try to do that in the clean way. First, we isogenize our stocks to be very careful about the genetic background. Second, we are not only making a single mutant, but we target all the gene family. For instance, we are now making a fly that will lack all the antimicrobial peptide. We know that they are supposedly to be important, but nobody has really tested their function. One idea beyond that is to know if effectors are also very specific. That means is effector is also shaped by specific pathogen. And this is actually underlined by a recent study that have suggested that diptericin, one antibacterial peptide, is just important against one type of bacteria, Providencia. That will suggest that there is not so much redundancy, but clearly different pathogens that shape also the effector response. So now to finish, I will show you a little story that we have done on prophenol oxidase. This was done by 2 PhD students, Jan Dudzik and Olivier Bingeli, with the help of a postdoc. So what is melanization? This is the most striking immune reaction in invertebrates, but it is not conserved in mammals. It involves production of melanin and reactive oxygen species. You see, it's quite spectacular. Insects turn black when they are infected. The role of melanization are multiple. Pathogen killings through the production of toxic reactive oxygen species, compound sequestration. People believe that melanin can sequester reactive oxygen species. And physical barrier, when you want to encapsulate the wasp and put inside all the toxic compound while protecting the host. In Drosophila, there are 3 enzymes which are involved in the production of melanization. They are called prophenol oxidase, and I will call them PPO. PPO1, which are produced by crystal cells, PPO2 produced by crystal cells, and PPO3 produced by lamellocyte. Here you see crystal cells. They are very spectacular because they contain crystals which are composed of pure form of PPO2. So this is a storage where you find a very large quantity of enzyme that can be released. So the cell disrupts in response to infection and release the enzyme. So I will go very brief, and I decided to skip most of the detail of our work, but our work has shown that each PPO has a specific function. And interestingly, we need to delete 2 of them to delete a particular function. So here you see melanization after injury. This is the hemolymphatic, if you want, the blood melanization that takes place when you challenge a larva. And we have seen that PPO1 and PPO2 are required. Only the double mutant show no melanization. PPO1 is the first source of defense, but PPO2 is a storage. It is found in the crystals. Interestingly, again, wasp infection, we observe that this is PPO2 and PPO3 which are important. Only when you mutate the 2, you find no blackening of the capsule. PPO1 has no role. So this shows you a kind of diversification and adaptation of those 3 genes to different functions. But now you can look to evolution, and there is lots of evolution studies in Drosophila, which has been, since Dobzhansky and others, a very good model for evolution. You observe that all the Drosophila species contain 1 copy of PPO1, but that the duplication between PPO2 and PPO3 occur in the melanogaster group that includes those 5 species. Here you find 2 genes. And now you can try to understand what does it mean, actually. And in fact, this mean of PPO3 arises with the invention of a new mechanism to fight against parasitoid wasps that involve lamellocyte. That means those insects are the only ones that contain lamellocytes, those large flat cells that can encapsulate the melanization. Interestingly, one of them, Drosophila seychellia, have lost PPO3. Why? Because Drosophila seychellia is a species that lives on a toxic fruits. It has a very specific niche, and in this toxic fruit, there is no parasitoid wasp. So, of course, you have relaxed selection, and they have lost PPO3 in this species. In other species, insects, Drosophila, have developed other mechanisms. In this mechanism, hemocytes fuse together to make very big machinery that can now start to encapsulate the parasitoid wasp. So here, now, by this type of approach, genetic systematic mutation, making double, single, and triple mutants, and by having this kind of evolutionary observation, we can now try to understand the logic of the fly immune system. So I will stop here. I've been maybe a bit briefer than I expected. This is my lab. This is what we are doing globally. We work a lot on spiroplasma, I mentioned a bit. We work a lot on immunity, and we are now a kind of extensive project to try to mutate at a large-scale level many of the immune factors. We are also a specific emphasis on the effectors, because I think we understand now quite well how we recognize, but to understand the effectors question, it's another, I would say, challenge for the future. And I thank you for your attention.